Okay, so welcome everyone again to the Green Area Hub webinar series. My name is Barry Crosby and I'm one of the communication officers for the Green Area Hub project. If you've been here with us before, you'll know that the Green Area Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERA-NETs, co-funds and self-sustained initiatives in the field of agri-food biotechnology. We started these breakfast clubs in October 2023, and we've had 5 very successful series so far. The first was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was Sustainable Food Systems. The fourth looked at precision agriculture technologies for a sustainable future. And the one that we just finished this month, in fact, was on plant health and protection. If you're interested in listening to the talks related to any of the topics from the series just mentioned, our recordings are now available to view on the Green Area Hub YouTube channel. Today, we're going to continue our new Breakfast Club webinar series, which focuses on the topic of animal health and welfare. So for the next 3 months or so, we will host a series of expert-led discussions aimed at fostering a deeper understanding of sustainable strategies for improving animal health, preventing disease, managing pests, and strengthening the connection between animal, human, and environmental well-being. Just a few housekeeping announcements. Please do feel free to type your questions in the Q&A and chat section, which I've enabled for everyone this morning. I'm going to do my best to address as many questions within the time that we have today, which is 1 hour. If at the end of the session you have a question for either of the speakers today, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Professor Jane Hodgkinson and Professor Peter Geldof, and we will open our session this morning with Jane Hodgkinson, Professor of Molecular Veterinary Parasitology within the Institute of Infection, Veterinary and Ecological Sciences at the University of Liverpool. A leading research scientist focusing on anthelmic resistance in Helminth parasites affecting both livestock and companion animals. Professor Hodgkinson's work leverages genetic and genomic tools to investigate the mechanisms underlying triclabendazole resistance in Fasciola hepatica, with the ultimate goal of improving parasite control strategies. In addition to her work on liver fluke, Professor Hodgkinson has a long-standing interest in drug resistance in equine parasites. She's actively collaborating with key stakeholders to develop evidence-based best practice guidelines for horse owners, veterinarians, and other anthelmintic prescribers. Professor Hodgkinson's research directly informs her veterinary parasitology teaching on the BVSC program at the University of Liverpool, where she brings cutting-edge science into the classroom with both passion and authority. Today, Professor Hodgkinson's talk is entitled Mapping Resistance Loci and Interrogating Mechanisms of Triclabendazole Resistance in European Isolates of Fasciola hepatica, which is part of the EU-funded International Coordination of Research on Infectious Animal Diseases, the ICRAD initiative. Jane, thank you very much for accepting our invitation to the webinar series. Again, you're very, very welcome, and thank you so much for joining us. I'm going to stop sharing my screen now, and you can begin sharing your presentation. Thank you very much.
Thank you for the invitation, Barry. And I will tell you about the work we are doing with Liverpool, but with multiple European partners under our recent ERA-NET. We're about six months into that project. So we have another 2.5 years to go. So it's a great opportunity to tell you about what we've done so far, but also the work leading up to that. And our remit was really to tell you within the area of anthelmintic resistance, which I've been working in for about 26 years, sort of cutting edge thinking that we have, and how that's kind of led to the sort of work that's currently funded under our ICRAD. So that's kind of where I'll go. So I'm sure many of you know this, but it's always worth just reiterating the significance of Helminth parasites to multiple different animal hosts, particularly ruminants and horses. And I'm going to focus primarily on parasites affecting sheep today, but it's important to appreciate that there are a number of resistant studies working on cattle parasites and equine parasites too. And if we look more generally at Helminth infections, and certainly they have a substantial impact on both the health and the welfare of ruminants. So we know that there's a long documented history of the impact that they have. And you can get different presentations. You can have subclinical disease, overt disease. We get acute fasciolosis through infection with Fasciola hepatica in sheep that will kill sheep. And along with measures to try and mitigate the impact of these parasites, the costs of these Helminth diseases to the livestock sector is substantial, running into the hundreds of millions, if not billions of pounds annually. It's difficult to put a final figure on the economic cost, but those economic studies that have been done show that it's a substantial impact. So there is this need to control these parasites, to mitigate the impact on animal health and welfare, but also to maximize productivity. And inevitably, the best way that has been sort of used to control these parasites is by treating the host with drugs. And I think some of the things that have changed very recently is around an awareness that many of these drugs can impact negatively on the environment. So that's another element of parasite control that's really being looked at, along with resistance. So in other words, we know drug resistance is occurring. We know we have to be careful how we use the drugs, but now we know they have a substantial impact on the environment too. It's really important that we only use these drugs as and when they're required. The rise of drug resistance has driven a lot of sort of good practice, or at least recommendations for good practice, to try and reduce the use of these drugs, therefore slow the development of resistance, but also just really to try and control this parasite in other ways that don't just rely on chemical products. We've known for the last couple of decades we really need to reduce the excessive use of anthelmintics, and the steps that have been taken to help end users, farmers, producers to actually use their drugs judiciously have been things like improving diagnostics, so we can target drug treatments. And in the case of anthelmintic resistance that I'm talking about today, to better detect resistance so that then we can employ the drugs or deploy the drugs in a way that they're going to be most effectively used. The problem of anthelmintic resistance, I'm going to focus just on these groups of parasites because they're really at the cutting edge of our understanding of the sort of the molecular mechanisms underlying resistance, are really the gastrointestinal nematodes. There's things like Haemonchus contortus and Tuladorsacea circumcincta that infect sheep, and the liver fluke Fasciola hepatica that infects sheep, cattle, humans, many different hosts. And these images just try and reflect the extent of anthelmintic resistance. I'll just put my pointer on. There we go. So this image here just shows obviously a map of the world, and it's coloured according to the different regions of the world. Haemonchus contortus is a parasite which is found in many different parts of the world, and these little dots here represent the studies. This is from a recent paper, the studies that have been carried out on different isolates of Haemonchus contortus. So you can see that drug-resistant studies on Haemonchus have been carried out all over the world, and that reflects the fact that you get infections there, and therefore you have to understand resistance much better, and that's why those studies have taken place. If we look at liver fluke and Fasciola hepatica, which is what I'm going to spend most of my time talking about, you can see that we have the colour coding reflects the number of cases of resistance reported. So you can see here many cases of resistance in the UK to a drug that's called triclabendazole, and you can see cases in South America over into Australia. So there are a number of different individual case reports of sheep infected with parasites that are resistant to the drug triclabendazole in many different parts of the world as well. And we've done extensive studies with farmers in the UK, trying to respond to their needs really, because acute fasciolosis, which is caused by the immature stages of Fasciola hepatica migrating in the liver of growing lambs, is a real concern for farmers in the UK, particularly along the western part of the UK where the climate favours its transmission. And we worked with farmers to basically take fecal samples before and after drug treatment to tell them whether they have evidence of, in this case again, it's triclabendazole resistance. And you can see here there's lots of little red dots. This is the northwest of England, and it's a big sheep producing area, and you can see that many of the farms there have examples where the drug is no longer effective because these red dots indicate cases of resistance. The first case of triclabendazole resistance was reported as far away as 1995, so before I started working on this. So we've known about it for a long time. And I think those practical ways of detecting resistance are along the lines of what I just described, these fecal egg counts pre and post treatment. So really, when I started, that's kind of where we were. We could detect it by taking these samples, these fecal samples, and doing sort of parasitology in the lab and looking for signs of eggs. But what's really happened in the last 25 years has been the advance of molecular approaches to try and understand the drugs, to try and understand the genes that explain resistance to these drugs. And I think it's worth just highlighting the complexity of these parasites. So again, sticking with Haemonchus and Teilodorsacea and Fasciola, which are really driving forward resistance studies within the veterinary helminths, because we have access to their genomes finally in the last decade. So Haemonchus and Fasciola are really complex organisms, much more complex than bacteria and viruses, and they have much bigger genomes to reflect that. So you can see the genome size of Haemonchus is around 285 megabase pairs. And with Fasciola, we were surprised. It was 3 times bigger than we thought it would be. It's about 1.2 gigabase pairs. So it's a huge genome. And what that means is when you're looking for the genes that are associated with resistance, you've got a very large genome to look through. It also makes sequencing very expensive because you have to sequence these very large genomes in order to get the sequencing data you need. So they're complex organisms to work with, but I think we've made significant advances in the time that I've been doing it, and hopefully we will continue to do so. So the earliest anthelmintic resistance studies in Fasciola and in things like Haemonchus and Teilodorsacea really relied on trying to select what we know as candidate genes. So making an assumption either by knowing a little bit about how the drug worked, or knowing a little bit about some important genes that have been associated with drug resistance in other organisms, and trying to assume that they may be involved, and then investigating their role in anthelmintic resistance. And for drugs like the benzimidazoles, some really beautiful work was done in the 1990s, and they had some really good candidate genes. And I think it's fair to say that the candidate gene approach did have some success, and I've got a slide on that in a minute. But I think what's changed with the advances of genomic studies is we can take these whole genome approaches now. So we have to generate our draft genome, and then we can compare the gene. If we resequence the genomes of resistant and susceptible parasites, we can compare them in a non-biased way. And therefore, by comparing them without making any assumptions, we can see which areas of the genome are associated with resistance, and then interrogate those genes within that region. So it allows you to come at it from a very different perspective. All these studies, both candidate gene approaches and whole genome approaches, and as I say, the whole genome approaches have really moved on the field in the last 10 years. All of them require a resource, a biological resource, of really well-defined isolates that are phenotyped for their drug sensitivity. So in other words, isolates that we know are drug susceptible or drug resistant. And it could be that sources of these have been long-maintained laboratory isolates that have been kept within individual labs, and they've been phenotyped and maintained over time. Or also, ideally, going into the current situation, going out onto farmland, collecting eggs, bringing them back, and testing them to see if they are drug responsive or if they are drug resistant. So identifying some novel isolates too. So all of those assays, and I'm just going to start with that, the assays to phenotype these isolates have to keep pace with the innovations we've made in molecular and genomic advances. And I think that's a really important point which needs really recognizing. Our genomic studies are really good, but we have to have things to work on, and we have to know that they are accurately phenotyped. So the different ways of phenotyping, and I'm going to lead into what we've been doing, but the different ways of phenotyping, you can phenotype in vivo. So you can take experimentally infected sheep, just treat half of them, and then sacrifice the animals, recover the parasites, and compare adult parasites from untreated sheep to treated sheep, and look at the genetic differences. Or in live sheep, you can take eggs taken from fecal samples from untreated sheep and compare them to eggs from treated sheep. So in other words, these eggs are a mixture of susceptible and resistant genotypes because the animals weren't treated, whereas the eggs from treated animals are made up of eggs that are resistant to the drug because they survived drug treatment. So there's a number of different in vivo approaches you can take. That's highlighting just 2. And then in vitro, I think, again, we've had advances in this area, but we have things like the egg hatch test, where you can take the eggs from fecal samples, you can expose them to the drugs in a petri dish or a tube, and then you can look at their survivability, so either their embryonation, their hatching, and if it's in the case of larvae, we can look at their survival. So we need these phenotyping assays in order to do these studies. And that was certainly the case. As I said, the candidate gene approaches have had some success. And just here now to name the drugs, the drug classes, the benzimidazoles. The benzimidazoles studies that were done first, because it's the one of the older drugs, they identified some candidate genes like beta-tuberin and P-glycoprotein to play a role in drug resistance. Levamisole candidates such as nicotinic acetylcholine receptors have been shown to play a role. And then it's more difficult with the macrocyclic lactones, potentially because they have multiple different genes involved in resistance, or at least that was the theory at the time. The candidate gene study suggested P-glycoproteins and glutamate gated chloride channels with some evidence for their involvement. But it's certainly true to say that these candidate gene studies, whilst they identified a few candidates, could not explain all the resistant phenotypes that we see. So the candidate gene studies were great. They were carried out on those phenotyped parasites. But if we then look at the advances that we've made with whole genome approaches, taking these same parasite isolates that have been well phenotyped and doing this whole genome resequencing and then comparing them, allowed you, as I say, to take that non-biased approach to identify areas of the genome that were enriched in resistant parasites. So otherwise, in other words, by comparing the genomes, you can essentially say, if I compare the genome of resistant and susceptible parasites, which areas of the genome are present in resistant parasites that we don't find in susceptible parasites, or at least are enriched in number. So in other words, they are the inherited part of the genome that are transferred into resistant parasites. And that's what the whole genome approaches have allowed us to do. But what's interesting is if you look here, we've got the drugs here. This is a control, so this is no drug treatment. This is for Haemonchus contortus in sheep. Control, fenbendazole treated parasites, levamisole treated parasites, and ivermectin treated parasites. So if you look at the fenbendazole one here, this is the chromosomes. Haemonchus has 5 chromosomes and an X chromosome. So you can see in the genome within chromosome 1, there are examples of, in fenbendazole resistant parasites, an area of the genome which is inherited by those fenbendazole resistant parasites. And if you look in a bit more detail in that region, it says here that that's actually a beta-tuberin gene. Now, if you remember from the last slide, beta-tuberin was actually identified in the candidate gene studies. So in this case, the genomic approaches are really beautifully aligned with the candidate gene studies to say, yes, certainly that gene is heavily involved in resistance, although it doesn't explain all resistance. And there are some other genes in here too. If we look at levamisole, there's 2 areas of the genome in chromosome 4 and chromosome 5. And again, there's some candidate genes here which were certainly implicated in candidate gene studies as well. So again, this is aligned quite nicely with those. So as I say, the candidate gene studies that have been going on prior to our having access to whole genomes did have some success in identifying those candidates. But macrocyclic lactone resistance, so a drug like ivermectin, this is always more complicated. And this is where the whole genome approaches really come into their own. You can see here there's an area of chromosome 5, which is inherited resistant parasites. And when we look at that and look at the genes in that region, there's one gene that stands out. It's called CKY1. And that had never been implicated in ivermectin resistance through candidate gene studies. So this non-biased approach allows us to really identify some potential candidates here. And for all of these genes, the role next is to go on and ask the question, what is their involvement in resistance, and explore that further. And then, because these are individual isolates we've looked at, look at how extensively these genes are found, or these areas of the genome are found under selection in different isolates of Haemonchus contortus in different parts of the world. And that's the work that's going on now. So the last 5 minutes or so, I'll tell you about our study. And this is on triclabendazole resistance in Fasciola hepatica. And it mirrors very much what's been happening. The Haemonchus work was running alongside our work, and we were taking a very similar approach. So it's really just describing that in more detail for you. In the UK, Fasciola hepatica is a really important parasite. We have farmers who lose a proportion of their growing lands every year due to acute fasciolosis. So it causes overt clinical disease. The subclinical impacts are multiple, both in sheep and cattle, in terms of reducing their milk yield, in terms of affecting their reproductive potential. So Fasciola hepatica is known to cause a lot of both subclinical and clinical impact on livestock in the UK and more broadly in different parts of the world. Along with its sister species, Fasciola gigantica, is found on pretty much every continent, I think, except Antarctica. So it's a very very prevalent parasite. It's economically important because it impacts on productivity of livestock in particular. And it's also a zoonosis, so it's recognised by the WHO as an emerging zoonotic parasite. In the UK, we've done a lot of work. There's a number of us that work on liver fluke because it's a primary concern to farmers. And what we know is that there's been an increasing prevalence. You can see this from this figure here. It fluctuates, but essentially, if we look back over time, in the last 2 decades, there's been an increase in the numbers of infections, an increase in the number of cases of clinical disease due to Fasciola hepatica. We also know that in areas of the UK that didn't typically have problems with transmission of Fasciola, they have seen cases of disease and cases of infection where they didn't historically. And we think that's due to our changing climate, shifting the snail, which is the intermediate host for Fasciola hepatica. It's allowing that snail to propagate within those environments because of the changed climate. In terms of the drugs that are available to treat Fasciola, because it's a big challenge for farmers to try and mitigate its impacts, there's lots of different flucicides that are available. There's benzimidazole, such as albendazole, and the benzimidazole, triclabendazole as well, that I'm going to talk about in more detail. But there's most of the drugs either target adult parasites. So with Fasciola, I haven't got a life cycle diagram, but essentially from the point of acquiring infection from picking up infective stages on grass, it will take about 12 weeks for the parasite to reach maturity to the adult stage. And it's really the first couple of weeks of infection, these juveniles here, at 1 to 2 weeks post-infection that cause most disease and acute fasciolosis. And triclabendazole is the only drug that targets those juveniles that are migrating in the liver. And that's why it's been the drug of choice and such a good drug. And that's why drug resistance to triclabendazole poses such a threat. So going back about 10 years, maybe even a bit longer than that, we started to take this genome-wide, unbiased approach to map resistance loci for triclabendazole resistance in Fasciola hepatica. And we did this by we have a snail colony at Liverpool, so we passaged isolates through our snails. We crossed in vivo, so we did a forward genetic cross where we took a susceptible parasite, a resistant parasite, crossed them through sheep, put them back through snails, put them back into sheep to allow the F1 to intercross. And then we resulted in a population of adult parasites that were either drug resistant because we treated the animals with triclabendazole. So this is a pool of adult parasites that are resistant to triclabendazole. And in untreated sheep, we had a pool of parasites that were a combination of drug susceptible and drug resistant parasites, because these animals remained untreated. And you can see here, this is just the number of parasites found post-mortem, but you can see that drug-treated animals had fewer parasites than the animals that were not treated. And that we would expect that because we've killed off the susceptible parasites that they were harboring. We also, because we ideally like to do this in a live animal, we've worked up a protocol to be able to take eggs recovered from sheep, either that are not treated with the drug or are treated with the drug. And this principle is the same. The eggs in the fecal samples of treated hosts are eggs coming from resistant parasites, whereas the eggs from untreated hosts are a mixture of both susceptible and drug-resistant parasites. And that gives us that comparison that we need to be able to do our whole genome comparisons of drug-resistant and susceptible parasites. And I have a very good statistician, Steve Patterson, who does the statistical analysis to identify areas of the genome in the selection. And that's what we're showing you here. This is essentially just the whole genome of Fasciola hepatica in a number of different scaffolds, because it's not quite contiguous into chromosomes. It's not quite as nice as we had for Helminth, but essentially all these represent different sections of the hepatica genome across here. And you can see in both our experiments, there's an area of the genome that lights up, that essentially is the area of the genome that is inherited by resistant parasites. So somewhere in this region of the genome are some genes that are driving triclabendazole resistance, because statistically they're overrepresented in resistant parasites. And we've had a look at this region in more detail. It's around 6 megabase pairs. It contains around 60 genes. So this is just showing the same, each of these little dots is just representing this region here. You can see the peak of the region sits here with these orange dots at their greatest. So we think the most interesting genes are lying in this region. There's about 25 of them that we're now exploring in more detail. But amongst them, there's an ABCB1, which is a P glycoprotein gene that was potentially implicated in candidate gene studies. It's known as a multi-drug resistance gene. So it's an interesting candidate. It sits slightly up with our peak, but we're still interested in it. then there's other genes, new genes that we haven't identified before, ADP robustylation factors, RAS-related protein, and fatty acid binding proteins. So we've got a number of gene candidates now that we can explore further. But where the equiderinet really comes in is we did all this work that I've just described in individual isolates from the UK, and even within the UK, it was from the northwest of the UK. So we need to ask more broadly about whether what we've seen in terms of a genomic signature, where these 60 genes are inherited by resistant parasites, how often do you see that in isolates from different geographic locations, either within the UK or more broadly across regions in Europe? So in other words, is there a single origin of triclabendazole resistance, or are there multiple? And that matters because if we're going to design methods to detect, molecular methods to detect resistance, we need to know how many of these different tests we'll need, because if it's going to be multiple origins of resistance, we're going to need multiple tests. And ultimately, in terms of pinpointing the gene that's involved in resistance, gene or genes that are involved in resistance, we need to understand better how these genes work in terms of driving susceptibility and resistance in parasites by using them in vitro drug exposure assays, which we're doing in collaboration with our partners in Queen's University Belfast. So this is the equiderinet. As I say, it only started in October 2024. The overall aim with all our partners is to improve the detection of anthelmintic resistance in the liver fluke, Fasciola hepatica, and mitigate the impact drug resistance has on ruminant health and productivity. And there are a number of different partners. We've got little images here of where we all are. It's myself, my colleague John Graham Brown, and Steve Patterson at Liverpool. Steve does a lot of the genomics and all the stats. John Graham Brown is a livestock vet, so he works a lot with the farmers. At Queen's University Belfast, we have Aaron Moore and Nikki Marks. Aaron has pioneered in vitro phenotyping assays and drug exposure assays for Fasciola. University College Dublin, we have Grace Mulcahy and Theo Duval, who have a long history of working on Fasciola. And then in Creavet, which is an SME, small medium enterprise in Belgium, we have Johannes Charlier and Alistair Antonopoulos, along with their industry partner, who's Francois Klein at Arcia. And then we have Yunus Emre Beyhan, who's working with us from the Van region of Turkey to understand more about Fasciola transmission and resistance in that region. And then we have an established collaboration with Georg von Somschel-Himmelsterner and Jürgen Krucken at Free University of Berlin. Again, they've recently identified cases of triclabendazole resistance in German sheep flocks. So this is just to finish, because I haven't got any results to show you yet, but I can describe where we're at. There's 4 different work packages, and they all feed into one another. We currently have a survey that we've launched that you may see out there actually in the different countries. We've had it translated into all the native languages, and that's currently launched and is being filled in by farmers to try and understand their use of different drugs to control liver fluke, including triclabendazole. And then we're going to do an analysis at the end and tie that in with some actual farm data where we do fecal count reduction tests. Obviously, we go to a farm, collect eggs before treatment, go back 21 days later and collect fecal samples again. And we can tell them whether they've had a significant reduction that meets the criteria for susceptibility. If they don't have that reduction, then they have cases of drug resistance. And then those eggs that we recover from the farms will go into exactly what I've just been describing. We'll then do whole genome resequencing from eggs, and we'll pick up signatures of selection with isolates from different parts of Europe. Some of the other isolates that are very resistant, we will put at Liverpool, and then we'll send them to Queen's University Belfast, who are currently doing gene silencing with RNAi in juvenile liver fluke. So we will be knocking down some of our candidate genes and looking at the phenotype of that knockdown. And along with that, Aaron is always improving their drug assays and phenotyping assays. So we'll take some of these isolates and we'll use it in our in vitro phenotyping platform too. So that's really where I'm up to. Maybe I'll get invited back and I can update you on our progress in due course, but certainly it's a great project to have, and we're really grateful for the equiderinet funding to allow us to carry out this work.
Jane, thank you very much for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Professor Hodgkinson at any point. Jane, if I may, just while we're waiting for one or two questions to come in, I was just wondering about the kind of the spread in terms of resistance in Europe to triclabendazole. Like, how widespread is triclabendazole resistance in Europe based on your findings?
So it's really interesting, Barry, because we know a lot about it in the UK, but when I asked people to be part of this consortium, we realized that it's there, but the extent of it is not really known because the focus tends to be on gastrointestinal nematodes. A lot of work will focus on that. And all the partners, the reason for them being involved is they wanted to know more about transmission, more about resistance. So that's part of this project, is to understand just how extensive resistance is. We do know there are individual cases, but the German partner is a really interesting example. We started work. I'm sorry about the dog barking, by the way. We started working with them about 4 years ago, and there had been no drug resistance studies done in certainly their sort of northern part of Germany at all for liver fluke, and they were really interested to know because they had a farmer who presented. He had 2,000, a flock of 2,000 lambs, and he'd lost 200 of them in a week due to acute fasciolosis. So that was their sort of way in, because then they had to investigate what he'd been doing, what drugs he'd been using. And then they sort of approached me and said, look, we've never really done a liver fluke study before and looked at drug resistance. So, could we work with you, take some of your protocols and do it? And they've now found, I think, they've sampled in the region of 30 or 40 farms, and they've found at least 5 cases of triclabendazole resistance. So I think we don't know because we haven't looked, and that's really part of this project. You would expect overt clinical disease, which I think is the most extreme end of resistance. So you could argue, well, if you're not seeing that much clinical disease, there's maybe not that much resistance, but actually we don't even know which flucosides people are using and how much triclabendazole is being used. So again, that was part of this project. We do know triclabendazole is being used in all the European countries. We don't know how effective it is. Ireland, Northern Ireland, and England and Scotland have all had many cases of triclabendazole resistance. But Belgium, Turkey, Germany, we have a Greek partner who might be joining us, far less information. So that forms part of the study. What we do know is if there's lots of fluke and triclabendazole is used, resistance will occur. It's just a matter of when.
Yes. Jane, that's fascinating. Just one last question just for me. I was just wondering about the next steps in relation to the project. So what would be the next step to turn this research project into something like tangible and usable in the field? So whether that would be, for example, developing like a diagnostic test or a new kind of anti-parasitic drug or treatment strategies and so forth.
Yeah. So I think all of those. So essentially, I think in an ideal world, we have to answer this question first of all of how commonly is resistance explained by a single gene? Because if it's multiple different genes, and you can have selection occurring differently in different populations geographically. So if that's the scenario, then it's not necessarily going to be realistic to develop one diagnostic, because you're having to develop a diagnostic to detect different genes in different geographic locations. Do I think that's likely? I think there's likely to be a major driver. There might be some subtle differences in different locations, but I think there's likely to be a major gene driving resistance in most contexts. So the argument would be, yes, moving to a platform like that. At the University of Liverpool, my colleague has recently developed a lateral flow pen-side test to detect infection. So we do have sort of the precedent there with moving to diagnostics. So I think we would like to develop. We're actually writing a grant at the minute to take it to that level where we could develop a sort of proof of principle diagnostic that would be a molecular diagnostic. But you can get them now that will work in a lateral flow format. They're sort of at the cutting edge of those sort of diagnostics, but we would like to try that. So yes, to the diagnostics, absolutely, that's something we'd like to do. Currently, the fecal count reduction test has all sorts of challenges. It's very time-consuming for farmers, had limitations in farmers wanting to engage with it. I think if we had something which was much easier to use for farmers, that they would definitely use it. So that's definitely in our next 10 years before I retire. Then when you were talking about all the time, we're emphasizing different ways to control this parasite. So rather than relying on triclabendazole, think about treating the transmission of liver fluke. Is obviously they spend time inside the snail. So treating them earlier in the season with a drug that just kills adult parasites, reduce egg shedding, reduce infection in the snails, reduce the number of metastaceae on pasture. Therefore, you have less disease. So yes, innovative ways that what we know about resistance is then driving different use of drugs to try and control the parasite differently. And then what was your last point? You said something else and I said all of them. I can't remember. You said diagnostics, management, and
new anti-parasitic drugs.
Oh yeah. So the way that that's probably working best, I think, is we've got we and others have drug-resistant isolates. So any new drug, any new compound that comes out must be able to target triclabendazole-resistant parasites. So our phenotyped parasites are often used in these screening assays because you need to know that the drug is going to work against resistant strains. So we have a project at the minute looking at spider venom toxins as potential because they're being used in other as pesticides and being investigated for those purposes. We're actually adapting that and looking at in vitro growing our parasites and then exposing them to these toxins and looking at those. So there's novel drug design happening as well. But as I say, the key to it is also testing those drugs on those novel drugs on things that are known to be resistant to existing drugs.
Sure.
Yeah.
And Jane, just one quick question, last question from our audience. Can resistant individuals develop tolerance to other drugs that have the same mechanism of action? Do you have a study on this?
Okay, that's really interesting. At the minute, because we don't really, when drugs are marketed, you don't necessarily know how they work. So we think triclabendazole has a unique mode of action. There's a number of reasons why we do. It seems to be very specific in the parasites that it can target. So fluke are very responsive to triclabendazole, but many of our nematode species are not. So we think there's probably something very specific about triclabendazole's mode of action, but we don't actually know how it does work. If you were to say amongst the benzimidazoles, which are purported to have a similar mode of action, all the benzimidazoles together, you have two of those represented, albendazole and triclabendazole, both will kill fluke. Triclabendazole will kill the juveniles all the way up to the adults. Albendazole will only kill the adults. But if you're saying they're classified together, those drugs, because they're benzimidazoles, so they could have a common mechanism of action, we know that triclabendazole-resistant parasites are still killed by albendazole. So in answer to that person's question, it's definitely possible. I think everything we're seeing so far suggests that of all the flukicides that are available, if they're resistant to one flukicide, another flukicide will kill them. Because Xantel, which is a different flukicide, will also kill triclabendazole. Whether very similar benzimidazoles with similar modes of action would do that, I don't know. That's a good question.
Jane, thank you so much. I think we'll just leave it at that. Unfortunately, we could go on and ask many questions on this fascinating research. Thank you so much for giving us such a wonderful presentation this morning. Okay, so joining us now is Peter Geldof, professor with the Faculty of Veterinary Medicine at the Department of Translational Physiology, Infectology and Public Health at Ghent University. An expert in veterinary parasitology with a research focus on the development of innovative control strategies against parasitic infections in ruminants and pigs. He earned his master's degree in biotechnology from Ghent University in 1998, followed by a PhD in the Laboratory of Parasitology at Ghent's Faculty of Veterinary Medicine, where he specializes in anti-parasitic vaccine development. Following his PhD, Professor Geldof spent 3 years at the Morden Research Institute in Edinburgh in the UK, advancing research into alternative parasite control methods. In 2006, he returned to Ghent University, where he now leads multiple research projects focused on parasitic disease in livestock. In addition to his research, Professor Geldof plays a central role in teaching veterinary parasitology, contributing to the academic development of future veterinarians with deep scientific insight and practical expertise. Today, Professor Geldof's talk is entitled Plant-Based Production of Anti-Nematode Vaccines, which is also part of the EU-funded ICRAD initiative. Peter, thank you very much for accepting our invitation for a webinar series. We're absolutely delighted to have you joining us today. So without further ado, the floor is yours. I'm going to stop sharing my screen and you can start sharing your presentation now. Thank you very much.
Well, thank you, Barry. First of all, for the kind introduction and the invitation of presenting the work that we've been doing in the last couple of years on vaccine development. Just see. So I hope my PowerPoint now is in full screen mode.
Yes, that's perfect, Peter. Thank you.
Okay, wonderful. Okay, so well, thank you for the invitation. As Barry said, my name is Peter Geldof. I'm working as a parasitologist at the Faculty of Veterinary Medicine at Ghent University. And quite a lot of our research has been focused in the last 10 to 20 years on the development of anti-worm vaccines in mainly for ruminant parasites. So in this presentation, I will mainly focus on the work we've been doing in the last 5 years in the context of an EU-funded project, together with colleagues from the Modern Research Institute, Leiden University Medical Center, and Wageningen University in the Netherlands, in trying to produce recombinant-based vaccines against nematodes in sheep and cattle. So as Jane already excellently introduced in the previous presentation, basically worm infections are omniprevalent, not only in the veterinary field, also in the human field. If you look in basically grazing ruminants, prevalence is basically 100%. So every animal that is grazing outside, cattle, sheep, goat, they will typically ingest nematode larvae or eggs on a daily basis and just be infected with different nematode species. Luckily, most infections will remain subclinical, and the effect of the infection is totally dependent on the level of worms present in the animal. In most cases, although it's subclinical, there will be an effect on productivity in terms of growth, milk production, egg production. So depending on which type of animal you look at, you will have an effect on this subclinical effect on productivity. And so to avoid these subclinical effects, farmers and vets typically will treat the animals several times a year with anthelmintics just to remove any worm infection that is present on that moment. Unfortunately, as Jane has focused on in her talk, the intensive use of these anthelmintics have led to the development of anthelmintic-resistant worm isolates, which have basically spread across the world and also in different host species. So in this table here, I basically summarized the different type of anthelmintics that have come on the market. So in the third column, you can see the year in which they were put on the market. So in the 60s and 70s, we had most of the benzimidazoles reaching the market. Later on, these were joined by the levamisoles, and then in the 70s and 80s, the highly successful macrocyclic lactones. In the last column, I basically listed there the year in which the first report of resistance against a certain compound was reported. And what you can see from this table, it typically takes about 5 to 10 years after market introduction of a new anthelmintic before the first reports of resistant worm isolates or treatment failure appear. And this is constant. So it basically tells us with any type of new compound we would develop in the future, the chance that worms will eventually develop resistance against it is almost like 100%. What is also an important take-home message from this table is that you can see the number of new compounds that have been launched on the market in the last 10 to 20 years has significantly decreased compared to the years before. There are two reasons for that. The macrocyclic lactones were extremely successful, with basically this negative side effect that the companies lost a bit interest in basically putting a lot of investment in R&D or looking for new compounds, because we assumed that the MLs would basically last forever. And second, there's also increasing demands in terms of regulation, safety, etc., before new compounds can be put on the market, especially for use in grazing animals in terms of ecotoxicity. And this basically leads us to the situation that we have very few new compounds reaching the market, and resistance against the compounds that have already been used in the last decennia is increasing and spreading further around the world. So we definitely need to not only focus research on trying to understand how resistance is developing and spreading, so referring back to the talk of Jane, but also thinking, well, how can we develop alternative control strategies against to control these infections in both animals and humans. And the use of anti-worm vaccines is a hypothesis that has been put forward in many different studies. And I've just included this output from PubMed here, just to give you an idea. If you just look at the keywords, nematodes and vaccines, you can see the first mentioning of that dates back to the 40s and 50s. And since then, there's like almost 2,000 papers who have been published on the topic of anti-nematode vaccines or the development of such vaccines. So I would not say there's like a huge worldwide interest, but still there's quite some substantial work been done on anti-nematode vaccines. Unfortunately, the reality is that at this moment, there are only two commercial anti-nematode vaccines on the market, which is the lungworm vaccine against Dictyococcus viviparus, which is branded under the brand name Huskvac in some countries. So this is used in cattle. It's based on irradiated larvae of lungworms, and it is still being sold and distributed in different European countries. More recently, there's the vaccine that has reached the market against Haemonchus contortus, which is an abomasal nematode species infecting sheep, mostly in tropical and subtropical countries, but now we see it spreading further also in more temperate climate regions. This is distributed and sold under the name Barbavax. Besides these two vaccines, there's basically none of them who have actually reached the market. And what is the reason for this? Well, to understand this, we basically have to dissect further the vaccine development process. And there are three important steps in this, in developing and putting a vaccine on the market. The first step is identifying the components that you're going to put into the vaccine. So it could be like crude worm homogenate, or it could also be a selected number of antigens, of which you would think that if we inject these or present these to the immune system of the host, the immune system will react against it and it will protect the animals later on against challenge infection. In the second phase, we need to think about ways of how to administer these antigens or protein extracts to the immune system, because we typically work with gastrointestinal nematode species, which are basically trigger or are in contact with the mucosal immune system. And then in the last phase, we need to be able to produce these vaccines in a sufficiently large scale and also in a cost-efficient way, which means that if we want to sell or have sheep, goats, cattle, or poultry being vaccinated, the cost of each individual vaccine should be realistic. If we look at the vaccine development process for nematodes, well, the biggest hurdle seems to have been step 3, is the possibility of producing these vaccines and these antigens in a sufficiently large way. And to further go in more detail, I would just use 2 examples of vaccines that we've been working on. So our group at Ghent University has been working on mainly on cattle nematode species, 2 worm species in particular, which is Ostertagia ostertagii, which is an abomasal nematode species, and Coperia oncophora, which is luminal small intestinal parasite. These are by most prevalent worm species in temperate or in regions with a temperate climate. So we've been working on development of 2 experimental vaccines against these species, and basically to summarize the project or the workflow for these vaccines. So we typically start with artificially infecting donor animals or calves with the parasites. After 3 weeks, we have to collect the worms, and after autopsy, we put the worms in culture, and we keep the worms in culture for about 1 week. And during that period, the worms basically survive in the culture medium and release and secrete antigens of which we assume that they will also release these or produce these when they're inside the bovine host. So these are antigens that we think will be important in the host-parasite interaction and could be important for the parasite itself. The 2 vaccine antigens we've been focused on, we call them ASPs, which is short for activation-associated secreted proteins. And these belong to a protein family that is unique to nematodes. All nematode species basically produce these proteins, and they seem to be released mainly by the parasitic life stages. So we do not exactly know what their biological role is, but we do know that they are released in quite high amounts during the infection, and that the host immune system is confronted with these proteins. We've been focusing on 2 of these ASP antigens. For Ostertagia, it's a protein of about 30 kilodalton in size. For Coperia, it's about double in size. When you purify these antigens and subsequently use them to immunize cattle, we see that we can actually raise a protective immune response against a challenge infection, against Coopera or Certagia. And the most important parameter for us in terms of protection is the secretion or production of worm eggs by the vaccinated animals. So, for example, with the Coopera vaccine, if you look at nematode egg secretion in vaccinated versus non-vaccinated animals, we can see we can reduce worm egg production by almost 90 to 95% following vaccination compared to animals who have just been vaccinated with the adjuvant. For Ostertagia, we also see this significant reduction in worm egg production, although it's lower compared to Coopera. And this is something we typically see with abomasal nematode species, is that the gastric tissue, the stomach, is less immunoactive compared to the small intestine. This is just a typical example of one vaccination study, but over the last 15 to 20 years, we've done dozens of these vaccination studies and always come to the same result, basically confirming the hypothesis that you can vaccinate animals against worm infections and it would protect them later on if they would graze on the field. So the problem, however, is when the way that we currently produce these vaccines is based on native protein material that we derive directly from the worms. And to have these worms, we have to artificially infect animals, which is ethically and also economically not sustainable to produce such vaccines on a large scale. So us and also other colleagues working on other parasite vaccines, we have to look in ways to produce these antigens in some kind of synthetic or recombinant way. And so people have been using bacteria, insect cells, pichia, or yeast to produce these antigens and then look at the protective effect of these recombinant versions compared to the native counterparts. And what we've seen, and also all the colleagues have been confronted with that, with any type of recombinant we produce, we failed to trigger, or at least we failed in the past to trigger a similar protective immune response in the vaccinated animals. In this slide, I show you two typical results. On the graph on the left is for Ostertagia vaccine, the one on the right is for Coopera. And what it shows here is the data of cumulative fecal egg output in vaccinated or control animals. So if you look for the Ostertagia vaccine, we have the cumulative egg output of control animals for about 2 to 3 months. If we vaccinate the animals with the native-based vaccine, we can reduce this with about 60 to 70%. However, if we look at the effect of antigen produced in pichia, so the equivalent of the native ACP but then produced in pichia, we see that in the vaccinated animals, we do not see any protective immune response or no reduction in cumulative egg output. And the same results or observations were also made for Coopera. This is a result obtained with the yeast-produced recombinant version, but we have similar results for bacterial or insect cell-produced recombinants. So what could be the problem with these recombinant versions? Well, we know that the recombinants we produce, independent of what system we use to produce them, they seem to be less immunogenic compared to their native counterparts. And in this slide here, I show you the results of the Ostertagia vaccine, where we compare the immunogenicity in terms of the cellular immune response and humoral or antibody immune response triggered by the native or recombinant version. The graph on the left basically shows the cellular reactivation of immune cells in the blood after vaccination. So in the x-axis is basically the weeks following vaccination, and the arrows indicate when animals are vaccinated with either adjuvant, native antigen, or recombinant antigen. And each week we collect blood and we basically re-stimulate the PBMCs, the immune cells present in the blood, with the antigen. And we can see if we re-stimulate the cells following vaccination with the native antigen, we clearly have a reaction of the cells. They recognize the antigen, they start to proliferate and divide. However, if we look at that similar response with following vaccination with recombinant, we see this is significantly lower. So the cells, the immune cells triggered by the vaccination with recombinant, react less to the vaccine antigen and also less to the native antigen. This is at the cellular level. If you look at antibody levels, we have the same effect. If you look at antibody levels triggered against the antigen in the mucosa following infection, so for Ostertagia, this is the mucosa of the abomasum. For Coopera, this would be in the small intestine. We can see that we only have a substantial secondary antibody response following infection after vaccination with the native vaccine, but not with the yeast-produced version. This indicates that structurally something is different in these recombinant versions and that they have different effect or activity on the immune system. And we've observed this several times for the Osteotagia and Coperia vaccine, but colleagues working on other vaccines against other parasite species have been confronted with exactly the same problem. And so one hypothesis that has been put forward for decades is the fact that these nematode antigens might have a specific structural protein folding linked to the presence of specific sugar residues present on the protein surface. So what are sugar residues or N-glycosylation, how we call it? Well, during the process of protein translation, expression, and the secretion of proteins, a lot of the proteins, and especially extracellular ones, are decorated with sugar residues. And these sugar trees are basically attached to the protein backbone, to particular amino acids. And these sugar trees are built up by individual sugar molecules and different kinds like fructose, galactose, mannose. And they're all indicated here with different basic symbols and color codes. So depending on the organism, whether it's a plant, an insect, a nematode, or a mammal, you will have different type of lichen structures present on proteins. And we know, for example, for the Osteotagia and Coperia native ASP antigens, we've done quite some work in trying to understand which N-lichen trees are present on these proteins, and they're indicated here in this figure. And what is particular or unique to nematodes are these fructose residues. These are these red triangles present at the bottom of this lichen tree. These are quite unique, and especially this one at the alpha-1 tree position, unique to nematodes and some other invertebrate organisms. They do not typically occur in mammalian N-lichens, and therefore they can be important in the immunogenicity of an antigen. So these are the N-lichens we've got on the native antigens. If you look at the N-lichens that are present on our recombinant Pichia versions, we have somewhat tried to mimic them, but you can clearly see that structurally they're still different. And so the question is, are these structural differences in terms of N-glycosylation really at the cause of having a lower immune response and protective effect with these yeast-produced versions? So basically, to address this, we had two questions. Do lichens present on nematode vaccines indeed play a role in the immunogenicity of the antigens? And are they essential to induce a protective immune response following vaccination? And if yes, well, how can we produce such recombinant glycoforms? Which system can we use to basically mimic these lichen trees? So for this, about, I think, 10 years ago, we got in contact with colleagues in the Netherlands and also with the colleagues from the Moorden Institute to basically develop a research workflow that is depicted here in this slide to address these two particular research questions. So the first step in this workflow, or the vaccine research workflow, is to start from native nematode antigens, of which we know they do induce a protective immune response when you vaccinate animals with them. At Ghent University, we focused on cattle parasites. The colleagues at the Moorden Institute, led by Tom McNeely and Al Nisbet, focused on nematode species infecting sheep. So our task here was to basically identify, purify, and identify native vaccine antigens that were subsequently analyzed by the group of Ron Hocke at Leiden University Medical Center for the presence of N-linked lichens, to basically see, are there sugar trees present on these proteins? What is their constitution? And in the second phase, to screen N-lichen microarrays, where we can actually see whether particular lichen structures are part of an antibody epitope or not. This information is subsequently used to steer the recombinant expression work at Wageningen University and the group of Ruud Wilders. And they've been working on an expression system in tobacco plants, Nicotiana, which has the advantage that you can basically play around with the glycosylation machinery of these plant cells. So you can knock out certain enzymes to avoid the introduction of certain plant-specific lichens, but you can also introduce, for example, nematode glycosyltransferases in the system to basically further tailor-made, for example, nematode-specific lichen structures. Antigens produced in this system were subsequently sent back to Ghent University or the Bourne Institute for further in vitro and in vivo analysis for their immunogenicity, and at the end, eventually for their protective effect. So we've been working mainly on Osteotagia cuperia, and the colleagues at the Morton at the abomasal nematode species, Taldorsasia and Circumcincta. So I will just go take some examples of results we got using this or applying this workflow. I will focus the presentation mainly on the Osteotagia vaccine, just as an example. So the first step was to basically show or deliver scientific proof that the N glycans present on the native ASP antigens are actually part of an antibody epitope, or that they play a part in the immunogenicity of the protein. So a technique that the colleagues at Leiden have developed for this is a glycan array, microarray. So it's basically a glass chip on which different glycan structures are spotted. And then this array can then be probed with antibodies from either native vaccinated animals, recombinant vaccinated animals, and you can measure the recognition or antibody binding to individual glycan spots. I just show you here a graph of a typical results we got. So at the bottom, you can see the different glycan structures that are spotted on the glycan array. And then the bar basically gives you the intensity of the antibody recognition. The green bars are antibodies present in serum from native vaccinated animals that had a protective immune response, versus the brown bar are antibodies present against the antigen in recombinant vaccinated animals. And what you can see is that there were quite a lot of glycan structures that were specifically recognized by antibodies of native vaccinated animals that were not recognized by antibodies present in animals vaccinated with the yeast-produced version. And if you look at the type of structure that are recognized, what is basically present in all of these structures are these alpha-1,3 core fucosa residues, which are already discussed in the previous slide, are basically invertebrate specific and can therefore be highly immunogenic. So the question is, how can we produce a recombinant version of this ASP that is also carrying this core alpha-1,3 fucosylation? So this is where the expertise of the colleagues at Wageningen came in. So as I explained, they've been working for quite a long time on a tobacco plant expression system, and they have basically been able to completely characterize all the different enzymes involved in the glycosylation machinery of these plants, and also have the tools to either knock out certain genes or introduce other genes from other hosts or organisms. So this gives you here a brief example of the different enzymes that were knocked out in their system and others that were introduced mainly from C. elegans, then to basically at the end have a glycoform that almost mimics the native glycoform as produced by the Osteotagia worms. So by applying this system, they were able to produce different glycoforms of the antigens that we're interested in. Just going to show you some examples here. So this is an SDA-based gel of the ASP antigen that was extracted from the plants or the leaves of these transformed plants. And on the top of this gel, you can see the different glycan structures that are present on these proteins. So they were able to produce an ASP version without any core fucosylation, so no red triangles here. One that solely carries an alpha-1,6, which refers to the position of this core fucosa, and then also one who has the alpha-1,3. If you look in the first step in vitro assays, looking at the immunogenicity or antibody recognition of these different recombinant versions, can clearly see, so the bar indicates the strength or the magnitude of antibody binding, that it's mainly the core fucosylated ones who showed an increased antibody recognition compared to the Pichia, and also the one who does not have any core fucosylation. So this was a first type of screening we performed with the newly generated recombinants before we took them into in vivo vaccination studies, as these in vivo studies come with quite a high cost. So a mixture of these two core fucosylated versions were tested for their protective capacity. In the first study, we combined it or compared the results with control animals, which are just vaccinated with the adjuvant, but also with a group of animals that were vaccinated with the native vaccine, of which we know induces a protective response. And we're very pleased to see at that moment that if you look at the reduction and fecal egg output in the vaccinated animals, that we got a similar effect in the recombinant vaccinated animals, similar to the native group compared to the quillae. So the dotted line here is the egg shedding in the adjuvant control animals, and the two lines here at the bottom are from the native vaccinated group and the Nicotiana vaccinated group. So we saw about 60 to 70% reduction in fecal egg output. So based on these results, we basically repeated the whole experiment. And then we just had two groups, one control group, and then again, a group of calves vaccinated with the plant-produced version of this antigen. And similar as in the first study, we again saw a significant reduction in fecal egg output in the vaccinated animals. So for us, we were very pleased to have these results because it was the first time that we were able to basically have a significant protective immune response in calves following vaccination with a recombinant version of this ASP antigen, which we did not obtain in the past with either bacterial or yeast-produced version, simply by mimicking the glycans present on the native antigen. To confirm at the end that the glycans play an important role in this, we took serum from these Nicotiana vaccinated calves and then rescreened the glycan array that we done in the first step of, or the second step of this research workflow. And we could now nicely see that by using these modified glycoforms produced in Nicotiana, we now were able to trigger an anti-glycan antibody response in these vaccinated animals, similar and even to a higher magnitude compared to native vaccinated animals. So this was clearly different if we compare it to Pichia vaccinated animals in the past. The years following on to that, we basically repeated the same workflow, but then on the Copiria ASP-based vaccine. So I just summarized it here on one slide. So in the first step, we actually showed that the native ASP antigen that we purify from the excretory secretory material from Copiria worms, that it actually protects calves against the challenge infection, reflected in a significant reduction in worm egg shedding in the vaccinated animals. We then looked at what type of N-glycans are present on this native ASP. We identified them through mass spectrometry. This information was then used to steer the expression work in the tobacco plants with the aim to basically form or produce glycomimics. And these recombinants at the end were then rechecked in vivo in cattle studies. And we could, again, similar as with the Osteotagia vaccine, show that we could trigger a protective immune response in the calves against the challenge infection. What is, however, important to notice, if you look at the levels of reduction we have in the recombinant vaccinated animals, although it's a significant reduction, it is still less efficient compared to the native version of the vaccine, indicating that there might still be some structural differences in the native versus the recombinant antigen that could be important in terms of their immunogenicity. So what are we currently focusing our work on? Well, basically trying to understand which glycoforms are exactly needed to trigger this protective immune response. Because if I show you the figures I showed early on, what type of glycoform is present on the recombinant versions, we typically show like one structure. But it's important to realize that if we look at the total presence of different glycoforms in the recombinants that are produced, that we're always dealing with a mixture of different forms there. And for example, here is an outcome of a mass spec analysis on the recombinant Osteotagia ASP, and you can see that the plant produces different glycoforms. They're basically all somewhat different from each other. The ones that we need are in there, but there's still quite a lot of other versions that we would prefer not to have in our vaccine, especially not if we're talking about batch differences, etc. So this is what we're currently focusing on, is trying to understand which glycan structure is really needed, and how can we further steer or optimize the expression system to really have the desired glycoform in the mixture. So by this, I come to the end of my presentation. I would like to thank all funding agencies that have been involved in this project. It's FWO for Flanders, or our part at Kent University, the Ministry of Agriculture, Nature and Food Quality in the Netherlands, BBSRC in the UK, and ICRAT for coordinating the whole funding. thank you for your attention, and more than happy to take any questions or comments.
Peter, that's super. Thank you very much again for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Professor Geldof at any point. Peter, just while we're waiting for one or two questions to come in, I was just really struck by the novelty of this particular project and the use of plants for developing plant-based approaches to developing vaccines for animals. And I was just wondering, could this plant-based approach be adapted for human vaccines, do you think? Or is it mainly just for veterinary and for agricultural purposes?
I think that the plant-based production system, if we talk to the commercial companies, this is not the type of production or expression system that they would typically use in large-scale antigen production, because it requires specific investment, etc., for growing the plants, harvesting them. For us, we more use it as a way to produce desired glycoforms of the recombinant versions, and then show that by modifying these, we can improve the immunogenicity and protective capacity, and then take that information on later to other expression systems to basically produce the version we really need, and then go back to, for example, yeast-based systems. Because if we really look at commercial vaccine production and purification, plants are not that easy to work with. There have been some commercial companies who've really used the plant-based system to produce commercial vaccine. Mainly this was in the COVID period, an interesting approach. But if possible, companies would prefer another production system just because it's quite expensive and difficult to work with.
Excellent. And what about the kind of your future research goals, Peter? I know you just touched on that a little bit at the end, but I was just wondering, what are your future research goals in terms of, like, is it refining the plant system, improving immune responses, or moving into trials? But I was wondering, could you maybe just expand on that a little bit for us, please?
It's to further basically refine or identify the exact glycan versions that we need to trigger the protective immune response, and then take that information into other expression systems to basically upscale production and retest those versions in vivo studies. So in the first couple of years, this is still, how we say, more academic or fundamental type research, but in the later phase, logically, this would have to be done in collaboration with commercial companies who have more expertise in the vaccine production. We are mostly focusing on Osteotagia and Coperia because in our part of the world, these are by far the most important gastrointestinal nematode species in cattle. But the same basically technology or workflow can also be adapted to other host parasites systems. So we already have the work on Teledorsagia in sheep, but that can be further expanded also in companion animals or cats, dogs, etc.
Fantastic. And Peter, just a question actually from Jane. She says, Peter, really nice presentation. In addition to the impact on egg shedding, how did the vaccines impact on numbers of worms?
It's a good question. For the vaccine against Coperia, the small intestinal parasite, there we do typically see a reduction in worm burden. For the abomasal nematode species, that is more difficult to obtain because, as I mentioned early on, the abomasum or stomach seems to be less immune active and seems to be more difficult for the host to really control worm burden or total number of worms present. What we see, a first sign of protective immunity, and that also happens in grazing animals, is that the worms are less able to fully grow into an adult worm and basically produce eggs. And this is also the type of effect we see with our vaccines. What is important, some people might say, okay, if you do not reduce worm burden with your vaccination, what's the point? But these type of vaccines, we have to look at it from an epidemiological point of view. If you put vaccinated animals on a pasture, if they shed less eggs, the pasture would become less contaminated, and in the second part of the grazing season, there will be a lower infection level on the pasture, and so they would less reinfect themselves. And this is something we've already tested in natural field studies with the Copiria vaccine, is that at the end of the grazing season, these calves, the vaccinated ones, basically had 80% less worms compared to non-vaccinated animals. So it's a different type of vaccine compared to antiviral or bacterial vaccine, where we really reduce pathogen burden immediately. This is different, so we basically impact with the transmission of the parasites.
And Peter, just lastly, one question just coming in, another question coming in from Tara. She says, "Hi Peter, thank you for the clear and insightful presentation. I may have missed it during your talk, but could you discuss why using C. elegans as the expression model has not been feasible?"
We years ago, together with people at Glasgow University, with Colette Britton, we've looked at that, and I think there's still some groups looking at that because it would be the most logical choice. You can keep C. elegans as a nematode, you can keep it grow it in vitro, in the lab, you can transform it. The problem is that at that moment we knew not enough about the actual lichen structures present on the Ostratagia ASPs or Copiria ASPs. And then to further steer the glycosylation pathway in C. elegans is not that straightforward. So the plant or the Nicotiana system was basically already further advanced for that type of use. And it has advantage that in a couple of weeks you can basically generate a new glycoform of the desired antigen you wish to produce. So at this moment, it's more handy, more practical in use compared to the C. elegans expression system. But the C. elegans system has a similar disadvantage compared to the plant-based system, is that to basically get this into commercial vaccine production is for many companies like an absolute no-go.
Peter, thank you very much. I think we'll have to leave it there, unfortunately. Again, just like Jane's excellent talk, I think we could go on for another half an hour easily with questions on your fantastic presentation. I would just like to thank once again our two excellent panelists who joined us this morning, Professor Jane Hodgkinson and Professor Peter Geldof, for giving us, I'm sure you'll agree, wonderful and insightful presentations and providing us with a great overview of their respective projects this morning. Our next webinar will be on May the 22nd, and I'll be sending the invitation out to all of you in the coming days. In that breakfast club, we will have Professor Javier Bezos, Professor in the Department of Animal Health of the Veterinary Faculty at Complutense University in Madrid, Spain, talking about improving diagnosis of tuberculosis in domestic ruminants. And we'll have Dr. Keenan O'Driscoll, actually, from the Animal and Grassland Research Innovation Centre here in Teagasc in Ireland, discussing the One Well Pig project, a framework to develop alternative high welfare pig production systems. Again, thank you very much to our panelists, and thank you everybody for joining us this morning and for your questions. We really hope you enjoyed the webinar as much as we did, and we hope to see you all again next time. Thank you very much, everybody, and have a good day.